Good afternoon, senior. Dr. Uday. Vanakkam. This is Dr. Kumaresan. Senior, I'm audible?
One second. Just cross check.
Yes,
Dr.
sir,
Kumaresan.
sir.
Perfect. Yeah, Kumaresan, how are you?
Yeah, fine.
After a long time. Okay,
Yes.
okay.
Yes. How the things are going on in Wellstech?
Yeah, everything's fine.
Yeah, great. So we are waiting for your lecture.
Sure.
Yeah.
Good afternoon, everyone. Welcome to the third session of FBR TFP 2020. We cordially invite our keynote speaker, Dr. N. Uday Prakash, Associate Professor, Department of Biotechnology, Wells University, Chennai. Our chairperson, Dr. K. Kumaresan, Associate Professor, Department of Biotechnology, Kumaraguru College of Engineering, Coimbatore. And our co-chairperson, Dr. M. Kripa Shankar, Assistant Professor, Department of Biotechnology, Panaryaman Institute of Technology, Sathyamangalam. I now request Dr. Kripa Shankar to introduce the keynote speaker. Over to you, sir.
Thank you, Aarti. So good afternoon, everyone. So I'm Dr. Kripa Shankar. So I have great privilege in introducing today's keynote speaker, Dr. N.K. Uday Prakash. So when I gone through his biodata, I came to know that he has versatile experience in the field of mycology. So I will make myself as brief as possible to introduce today's keynote speaker. So to start from his educational qualification, he has completed his UG and PG in the field of plant science and completed his PhD in the field of aerobiology from University of Madras. And he is specialized in the fields of aerobiology, microbial biotechnology, herbal biotechnology, bioremediation, and bio-nanotechnology. And currently he is having a position as associate professor at Wells University in Chennai. And previously worked at World Tech Engineering College as associate professor, and also at New College and Alpha College of Arts and Science at Chennai. And he is also heading as director R&D at Marina Labs, Chennai. And previous to that, he has worked at Omni Environmental at USA as manager, and also at IAQ Laboratories at Singapore as director. And he has also got experience as postdoctoral researchers in different industries. And to add a few more distinctions, he got renowned awards like Inspirational Associate Professor Award, awarded by DST, Government of India, and Fellow of Indian Association of Applied Microbiologists in the year 2019. And he has also got Young Scientist Award in the year 2013. So right now we have an eminent speaker among us, and the session is going to be very useful. And I think we are going to get the informative speech. Sir, the dais is yours. Thank you.
Thank you, Kripa, for your introduction. Yeah, before I start, my thanks are due to the organizers of FB or TFP 2020. The convener, Dr. K.P. Kannam, the chairperson, Dr. Kumaresan, and then co-chair, Dr. Kripa. Thank you for providing me this platform to share my research experiences. Right, in this lecture, what I'm going to say is my travel in the field of aerobiology, specifically from the tropical country. India being the tropical country, it differs when we are comparing with that of temperate countries. So that is the unique advantage of learning aerobiology in India. It's titled in general as airborne fungi in different intramural and extramural environments. Yeah, what I'm going to discuss is just an introduction about what aerobiology is, because many scientists in India are not aware of this particular field. Only rarely, most probably, even with Indian Aerobiology Society, we have around only 300 members. So the field needs to be propagated in a well-mannered way. So let me give few introduction about aerobiology. Later, I'll get into the extramural environment and intramural environment, how the microclimate are playing a role. Then what is the current scenario happening? Whatever the thing I'm presenting right here, this are all my experiences from the year 1995, because that is the year I joined Professor Vittal's laboratory for my studies related to aerobiology. Right. In general, bioaerosols can be termed as any particle belonging to bacteria, fungi, actinomyces, fern spores, pollen, algae, plant cells, insects, mites, protein, enzyme, antibiotics, and even toxins when they are suspended in the atmosphere. And you are the one who are going to read about that, then you are termed as an aerobiologist. And in general, yeah, Dr. Kannan, any query? Dr. Kannan. Right, let me proceed with. So these things, when it is suspended in the atmosphere, they are termed as bioaerosols. Majorly, this involves the pathway of this is too much important, because any particle to become into air, they need a source, then a very good releasing mechanism. The ability for these particulates to suspend in the atmosphere, in other words, we can call it as flight efficiency of the particle. Then where it is getting deposited, and even after depositing, what is its impact? And this impact depends upon the surface or any organism where it is having a major role. In each and every stage of all these major pathways, majorly environmental factors plays a role. Typically temperature, humidity, moisture, and even rainfall. So these are all the things which are playing a major role in the pathway related to aerobiology. Here what you're seeing is just a fungal spore released from its conidia. In general, we can call it as a dispersal. So for any fungal spore to become an airborne, nature, it needs a source where the spores can be produced. Then the capacity for that specific fungal spore to be buoyant in the atmosphere. When it is going to produce an impact, that needs to be in significant number. So these three are the major criteria which are required for any fungus to be significant in the atmosphere. Why we are studying it? Specifically, always the human used to study the thing when it is having an impact on human race. These fungal pollutants, they are attacking the human race in three-dimensional way. Typically as similar to trident, it is affecting as an allergen, as a pathogen, and as a toxin. So it has a major significance in it. Further, the size, when you are checking on to the size of the fungal spores, it's falling between 1 micrometer to 50 micrometer in size, majorly. So these size particulates are having a capacity to enter your respiratory system. It enters through the bronchi, reaches the tertiary bronchi. Finally, if the spore size is less than 3 micrometer, they have the capacity to reach your alveolar zone. So this is what the significance of the fungal spore size is. So keeping that in mind, all these characters are exhibited by the fungal spores. So atmospheric fungi are important to study about the knowledge related to the presence of fungi in any environment is significant, right? With that, yeah, coming towards its impact, human diseases, as I have discussed it, agriculture, when it is falling on the plants, plant diseases are possible. Forestry, even they have the capacity to destroy the forest itself. Generally, you can call them as a pollutant of air. When they are falling on with even a library or any book or any deteriorating stuff, then they function as a very good deteriorators. Even they cause animal diseases, then they're also altering general ecology and phytogeography. So even when you are coming across any forest where trees are present in abundance, an infection of fungi can completely wipe out those trees, right? So those are all possible when the fungi are becoming airborne and its impact is greater. This is just a slide to create an awareness about the presence of fungal spores in the atmosphere. This particular slide, I have trapped it in US, particularly in Austin during winter time. This is the cocktail slide, which a microscopic picture, which shows you the presence of Alternaria, Clarosporium, Nigrospora, Periconia, Cercospora, Curvularia, Clarosporium in plenty, Aspergilli and Penicilli. So like that, plenty of spores are available in this particular single microscopic shot. So just think about the atmosphere, then how it will be filled with the spores. Right, medical importance. They are having the capacity to cause obstructive bronchopulmonary aspergillosis or mycosis, allergy, asthma, bronchitis, BRI in the sense it is called as building related illness. These things are majorly reported from Singapore. Apart from that, most of the industry where the buildings are considered as airtight, you can see people reporting BRI. So in temperate countries also you can have this prevalence of this particular disease. Conjunctivitis, mostly the eye irritation. Then few of the organisms which causes a cryptococcuses, that is cryptococcus infection, histoplasma, inhalation fever, that is flu-like symptom because of the fungal allergen. Then opportunistically, they can also cause infection. Toxic effect, actually I removed a couple of slides in this case because everybody, even you can Google it to know about the toxin produced by the fungi. Plenty of Aspergillus species and Penicillium species are having capacity to produce toxin. Here I've given Fusarium, Penicillium species, Pithomyces chartarum, and Stachybotrys chartarum. Stachybotrys, why I've highlighted here is in US, they are too much scared about the presence of Stachybotrys because it is completely nicknamed as toxic fungi. So when Stachybotrys is present indoor, they need to get a certification from the federal agency about the clearance of this mold in that particular building. Further, in apart from physical presence, they also produce the secondary metabolites, which are volatile. You can call them as microbial volatile organic compounds or mycologically produced volatile organic compounds. So they are also important. Right. This particular slide showing different type of samplers because whatever the result you are reporting after studying the atmosphere, that is completely depending upon the sampler. Again, each and every sampler is having its own advantage and disadvantages. First one, you are seeing liquid impinger, cost is economically cheaper. Maybe in India, it may cost around 4,000 to 5,000 rupees. Second one, Anderson sampler. Even in that Anderson sampler itself, you can see the cascade impactor, which are designed with 6 stages, 8 stages, and 10 stages like. Then the next diagram you're seeing is the 2-stage Anderson sampler. These are all costly and considered as a gold standard equipment in monitoring airborne fungi. Then the next one, RCS, labeled as RCS, that is Reuter centrifugal sampler. Here the advantage is it is battery operated. So most of the indoors, there is no need for you to carry a specific battery along with that. So simply carry this particular sampler, get into the indoor, take the sample, and you can easily come out, right? And those are all designed for monitoring culturable fungi. There are plenty of spores which are non-viable in nature, but present in atmosphere where they have the capacity to cause allergy. So to get the overall picture of the presence of fungal spores in the atmosphere, you can use the other samplers like biosis, advantageous one, aerosol, use and throw. For a single sampling system you can use, and then after monitoring, just you can dispose the sampler. Then the Cyclex. Cyclex, again, multiple used sampler. Burkhardt personal sampler designed by UK Rothamsted Station. Luckily, I have all these sort of samplers with me. When I Returned back from US, in India, we have procured all these type of samplers, and we are reaching this particular aerobiology sampling unit. And my laboratory is richest in India, related to aerobiology is concerned, right? Now, let me classify the area of study towards airborne fungi. Generally, they are classified into intramural and extramural environment. Intramural in the sense, when the sampling site is surrounded by walls on 4 sides, completely sealed area, you call them as intramural. Example, home, hospital, hotel, industries, places of worship, educational institution, animal houses, even transport system, malls, theaters. So when the environment is enclosed with wall on all sides and completely sealed, you call it as intramural or indoor environment. When there is no limitation, then there is no boundary, then you call it as extramural or outdoor environment. Example, garbage dumping site, vegetable markets, highways, agricultural fields, even a city, open city, public and other public environments like playgrounds, everything falls under extramural environment or outdoor environment, right? Here we are going to travel for a few minutes related to a few different areas. Maybe I may be skipping few slides even because I don't want to share much of data which may be boring for you, right? Yeah, let us travel into outdoor environments. That is also called as extramural. Here you are seeing that our sampler is in operation in traffic signals of Chennai. Then the tea estate. You can enjoy this particular environment to monitor the plant pathogens which are suspended in air. Just this picture, why I have showed us, it possesses a uniform plantation. So to study about the airborne nature of the fungi in this thing will serve you about forecasting the diseases, plant diseases. So that is the other major group of aerobiology, where plenty of plant pathologists are working on airborne diseases of fungi in agricultural plants. Here the farmers, just I want you to recall about the source what I said. Here you can see tons which are shattered here and there. Then just think about the flora, that is the fungal growth in this particular environment. Apart from fungal growth, here methane is emitted and other volatile organic compounds are also present. So as a cocktail, when you are inhaling or when you are serving in this particular environment, your health is at risk, right? So what we got in this particular environment, you can see that our sampler is in operation in the middle of the cattle sheds. So the top dominant fungi is Aspergillus flavus, which has 124 colony forming units per cubic meter of air. Cubic meter in the sense that is nearly 1,000 liters of air. So when you are inhaling 1,000 liters of air, when you will inhale, when you are staying in an environment for 2 hours and 48 minutes, there is a chance of you inhaling 1,000 liters of air. So if you are staying in this particular environment for 2 hours and 48 minutes, then your inhalation capacity of fungal spores is Aspergillus flavus 124, Aspergillus niger 89, Penicillium citrinum 41, Rhizopus stolonifer 39, Aspergillus versicolor 25. So of course the total may differ, right? Let me move on to, we have also published it as a paper in Czechoslovakia mycology by the year 2003, but this are all related to airborne thermophilic fungi. What we got? We have got a total average of 128 CFU per cubic meter air. Thermophilic fungi are concerned, they grow at high temperature, that is near 50 degrees centigrade, right? We are getting on to the next environment, that is vegetable market. Again, think about the source, the moisture, right, available in just that particular environment. Then propagation of fungal spore in this environment is too high, so plenty of fungal spores will be there. And think about the people who are working in that particular environment. Here, just see the number. Aspergillus niger alone, nearly 1,521 colony forming units per cubic meter of air. So your lungs will be filled with Aspergillus niger. followed by flavors and rhizophores. We have published a paper on pollution research related to the presence of airborne thermophilus fungi in the vegetable market, where it has given 227.14 colony forming units per cubic meter of air, that is the thermophilic ones. Right. This particular area you may be enjoying. These are all the unique nature of India. The markets right here, the fish markets, they are open, and then you can see that how the water are getting splashed here and there on the surfaces of the people who are walking and even who are coming there to purchase. The beauty of fish market, how it is in India. So these are all unique, right? Our sample right here is the impincher. We are using the liquid impincher sample in this particular market. What is the colony forming units we recorded? It's 236 colony forming units of fungi per cubic meter of air. Here, why the number is low? The moisture present, the water present in that area, that is not allowing the fungal spore to become dry and airborne. So that is why we are getting a lower number of colony forming units in this particular environment. I've also worked with bacteria, but as it is, the conference is out of title related with bacteria. I'm not presenting that particular data right here. Bacteria exceeds nearly 15 folds of this number. The other beautiful environment, particularly for biotechnologists, this is a cocktail of industrially important fungal presence environment. The fungi which are present right here have the capacity to produce the enzyme like cellulase, protease, lipase, everything, whatever you want that is present in this particular environment. Biotechnologically important, you can see that the lorries are carrying the garbages from the city. In Chennai, nearly 4,000 tons of garbages are generated in a day. So everywhere you can see that garbage is dumped. This particular environment is filled with garbages, and we have started studying this particular environment for 2 continuous years. What is the thing we got? The colony forming units exceeded 1,712 colony forming units per cubic meter of air. The dominant fungus, it's Aspergillus fumigatus. Why the Aspergillus fumigatus is dominating here? They have the capacity to grow from the temperature of 20 to 55. When you're taking the mean temperature, that is 37, which is very much optimal for the fungus to grow, which is your body temperature. Coming to the size of the fungal spore, it is less than 3 micrometer. As I said, they have the penetration ability of your lung, and successfully they can invade your alveolar zone. Just think, they have the capacity to reach your alveolar zone. The body temperature is optimum for the fungus to grow. So when they start growing, your lung capacity is reduced, then the disease, what you call it as obstructive bronchopulmonary aspergillosis. So think about the people who are serving in this particular environment. We have published a paper related with the presence of thermophilus fungi in this particular environment. On an average, what we got is 821 CFU per cubic meter of air, thermophiles alone. Coming towards, this is the paper what we published in Indian Journal of AIRBORNE by 2003, where you have already seen that 1,700 CFU per cubic meter of air was recorded in this particular environment. And the beauty of aerobiology is wherever you want to do research, just you can float and do similar to that of air. So we are very much interested to learn about the airborne fungi in Pichavaram forest. You can see Dr. K.P. Kannan, the convener of this particular conference, and Dr. Sampath Kumar along with me. Just we enjoyed this particular trip for studying the airborne fungal spores in Pichavaram zone of Chidambaram. Right. Our sampler in operation. You can see we have used again a liquid impinger right here, and we have recorded a lower number of fungi. Whatever the fungi we have recorded, just think they are not from soil. Mostly they are all emanating from the surfaces of the plant. Either it may be a pathogen or a general phylloplane infecting fungal spores. Right. And we have also thought about, okay, why don't we conduct a study related to coastal zone, as we are situated next to coastal zone. So here when you're seeing, this is the picture, what we have taken it from the Mamallapuram, which is a world archaeological important site. The Sun Temple belongs to 14th century. So there we have started our study. Then till the end of Chennai, nearly covering around 58 kilometers, every 1.5 kilometers, we did the sample, till Marina. So what you are seeing is the coast of Marina, which is second longest coast in the world. So there we have also studied about the presence of airborne fungi. The beauty is the fungal spores recorded next to the coastal zone is less than 20 colony forming units per cubic meter of air, starting from Mamallapuram to Marina. So when you want to walk, just trawl through the coast so that your lung cannot expose to much of fungal spores. So that is what the information what I am trying to give for you people. Right, let me get into the dissection of Chennai in different zones, and then I may be skipping few slides right here because it may be, it's a complete dissection of Chennai. This is from Kidpak, Niger, 100 CFU, flavors 89, Clarosporium 40. Next, this is from Rayapuram, Niger again dominating, followed by flavors, but third dominance is Aspergillus versicolor. Georgetown, flavors is dominating rather than Niger right here, followed by Niger, Clarosporiatus. Tharamani, Niger goes back, second position is occupied by Clarosporium, third by flavors. Gindi, Niger dominant, followed by Clarosporium, third flavors. Perungudi, Niger flavors, but third is Aspergillus fumigatus. So think about the zone. Why I'm highlighting this is, again, this is next to the garbage dumping site. Basin Bridge, Niger flavors Clarosporium. Mylapore, Niger, think about the count of Niger right there, 350, followed by flavors, and Rhizopistolonifer, again Syncephalastum racemosum. These two are moisture-loving fungi. So atmosphere of Chennai, when you are studying about 172 square kilometers, you are supposed to dissect them into different environment and start studying. That is what this particular study conveys about. Crumpet, Niger flavors Clarosporiatus. And fourth one, when you're checking, it is Monilia cytophyllum. This particular environment is colonized by the leather tannery units. Most of the leather are infected with Monilia, so the spores are suspended in the atmosphere. So even by using the air monitoring, you can say what is the significance of that particular environment. Right. Then we have published it as a paper, Aerial Spectrum of Fungal Biopollutants of Chennai. Nearly we recorded 81 species belonging to 32 genera. The dominance is Aspergillus with 19 species, as it is a tropical country. Whereas mostly in temporal country, over there, everything was dominated by Penicillium. So that is what the difference. When I did the same work in US, Penicillium started dominating. Whereas in India, it is Aspergillus dominating. Third, here it is followed by Curvularia, whereas in US it is Clarosporium. So for every geographical zone, the airborne fungi used to differ. So we need to be careful when we are analyzing the atmosphere of that particular specific environment, and it differs, so that the area is wide open for the aerobiologist to probe, and you can enjoy with the results, the different sort of results what you are getting. That is the beauty of this particular field. And why we concentrated on Ambathuras, just it is highly industrialized environment. So we started studying Ambattur as a mini site. We followed it with that of Manali too. In Manali, we got very lower number of fungal spores. When we are again checking it, Manali is having plenty of chemical industries. So these chemicals are not allowing the fungi to be viable or to become airborne. So that is what our conclusion related to Chennai, studies related to Chennai is. This other beautiful site. This is Neyveli Lignite Corporation. Myself, along with the Periyar University, we had a collaboration with Dr. Arvind, and we studied the air flora of a near lignite mine. Then we got only 260 colony forming units per cubic meter of air. Why again only 260? Just look into this photograph, you cannot see any vegetation, right? So it is not supporting the growth of or the absence of sources or the growth of fungi in any of the surfaces. So that is what we... Hello?
Senior, sorry to disturb you. In between there is a voice, background voice is there.
Okay.
I don't know.
Yeah, is that okay?
Yeah. Thank you, Senior.
Yeah. So over here, you can see that there is no organic matter available, so that the fungal spores recorded here is only low, that is 260 colony forming units per cubic meter of air. Right. So far, we have seen about the outdoor. Now let us peep into the indoors, how they are going to be. You'll be enjoying this particular mall. Whenever you want to visit malls, you will be having some happy stuff. You want to do window shopping, right? So one of my students has studied nearly 10 different malls of Chennai. Right now, I am unable to reveal the results because the paper yet to be published. Then the theaters, we have selected 25 different theaters in Chennai, and the study has been conducted. Again, the results are at stake, just we need to publish it. Then, coming on to the indoor buildings, right? Most of the IT and IT related enhanced business building, they are considered as airtight. Why? There is no external air transaction. They are completely closed, centrally air conditioned by AHUs. So the people who are occupying this particular building, they are most probably reporting sick building syndrome. When I worked in Singapore, most of the people are reporting such a thing. They are continuously reporting sneeze and cough throughout the year. It is not related to any climatic change. Whereas whenever they are getting into the building, they are getting this sneeze and cough. So it is clearly visible that it is because of sick building syndrome and building related illness or the general complacency, what is reported by them. After studying the indoor mold spectrum, we have presented it, published it as a paper. Then over there, we have taken all the sources even, right? Tunnels. This is a beautiful environment. Or you can call it as underpasses. This is just an underpass, pedestrian underpass present in Chennai, right? The people used to wade through to cross the road. So our interest is, it is completely different. It is sunken. Even external air exchange will not take place in this particular environment. There will be a depot. The humidity changes, moisture level changes, temperature changes. So we want to know what will happen in this particular underpasses. After studying it, we have published it as a paper just a couple of weeks before in AIRBORNE. What we recorded, on an average, 157 colony forming units when compared with the outdoor air of 114. So indoor is promoting the growth of fungi because of the moisture and few of the pedestrians throwing the garbages, and few petty shops are also present when you are going through the underpass next to marina, right? So these are all the conditions which are responsible for the presence of indoor fungi in underpasses. Guinea pig house. Here the environment is mostly 24 degrees centigrade. Plenty of organic material like the animal urinals, the straw material, that is the bedding material, the dung, everything is organic. So they are functioning as a beautiful source for over here for the propagation of microbes or the fungi to grow in this. Apart from that, the smell of the urine, that is emanating ammonia, everything again as a cocktail posing a health hazard for the animal caretakers. Similar situation in rabbit house, and we have presented it as a paper. Over here we have got an average of here the domination of Aspergillus fumigatus by 125 colony forming units. Food preparing unit, we have studied, but results yet to be revealed. This is the back-free. Couple of years we continuously monitored, results yet to be revealed. Even you people can work with the hospitals, the indoor air quality or the fungal quality or any microbial quality, how good your hospital is, or how good your intensive care unit is, because you are not allowed to see the patient. But how come we know the quality, air quality in ICU is good? So that is the need for us to study. And this is the mortuary area where again we need to explore more. Just we did a preliminary investigation in mortuary belonging to one of the state, a UT or state, it's Pondicherry, right? My students did their project over there, right? Then coming on to indoor, mostly you can see the sources, the restroom, the wall, even the hiding places whenever we play hide and seek. Then we have conducted a study on asthmatics of Chennai. Around 100 asthmatic houses were evaluated for the presence of fungi. Here we have reported only about the thermophils, where we recorded nearly 2,000 colony forming units. The other environment is, it's quite interesting, AC buses, because when the people want to fly, when they want to go through one area to other, they use this particular transportation. Again, they are going to spend nearly minimum of 8 hours within the bus. So we have conducted a study on the prevalence of fungal spores in 50 different air-conditioned buses flying from different areas. We have recorded nearly 38 species classified under 21 genera. On an average, nearly 13 colony forming units we have recorded within the buses of air conditioning. And this is the rapid transport system, RP or MTS. So here we have conducted the study, the result yet to be revealed. So these are all the area where you can probe, or even if you are interested, you can probe onto the flight of what is the microbial quality or the fungal quality present indoor of the flights. One special occasion, how the fungi, airborne fungi behave. This is a beautiful celebration by the Indians called as Bhogi. What we do, just we will be firing the organic material, the waste stuff, so that it generates a lot of particulate matter. Although it is religious, my concern is why we are doing it. And as an aerobiologist and aeromycologist, my concern is what will happen to fungi. My conclusion is when I'm studying the same on the control days, it is more in number. On bogey days, the colony forming units are getting reduced. So this is what the conclusion, what I got from studying nearly 3 consecutive years we have studied to know about the impact of bogey on airborne fungi. The result yet to be published. And what will happen during Diwali? Plenty of crackers are busted. Even the Supreme Court and High Court are coming in and playing the role, asking us to reduce the hours of playing crackers. So my question is, as an aerobiologist and aeromycologist, what is its impact on airborne fungi when we are celebrating crackers? And we published a paper in Helion stating the impact of firecrackers on airborne microflora. So bacteria got reduced, however, the fungi are concerned not much reduced. What will happen when solar eclipse is happening? Also, we haven't published the paper yet, just I want to say, when annular solar eclipse is happening, you can record there is a drop of 4 degrees centigrade of temperature. So there is a sudden drop in temperature. So the airborne fungal spores are low in number when the solar eclipse is happening at its peak. We have studied a couple of solar eclipses and the results are yet to be published. And what will happen during lunar eclipse? There is no change at all, because it is not going to have any impact on the temperature or anything, atmospheric temperature. And we recorded there is no change in any of the fungi. Again, we have studied 3 different lunar eclipses to know about the consistency in our result, and the results are yet to be published. Right. So far, what you have learned is, what is airborne fungi? In general, how it can be studied, what is its human impact, and how it is behaving in indoors and outdoors. Right now, what we are venturing is, what are the inventions by my laboratory in the field? UAVs, unmanned aerial vehicles, they are widely used in developed countries, mostly US, Australia, and UK. They have started using UAVs to monitor the airborne flora. Whereas in India, there was no study conducted so far. But as an aerobiologist, my concern is why not we imply that. So our laboratory is the first one to bring in UAV with the collaboration of MIT, Chennai, to bring in UAVs to monitor the airborne fungi. What you are seeing is that a couple of cells attached to a couple of both sides of the wing, and it is under flying condition. You can see the close-up view of our aerosol cassette. And we have presented it with a particular TV channel, Times Now, stating that as it is a wonderful invention, they interviewed us to know about the details related to UAV in usage of UAV in airborne monitoring. And we have published it as a paper. So we are the first one to introduce UAV to monitor airborne fungal spores in India. Later we thought there is a drawback in using fixed wing UAV. Is there any development? Right now we are working. Highlights, this particular quadrotor is having hovering benefit. Similar to helicopter, it can stand. We are the first one to use quadcopter in the world to monitor airborne fungi. This particular quadcopter has an ability to monitor physical, chemical, and biological aerosols. This is how the quadcopter, when we are designing, you can see, we are fixing the wings. And you can see that our quadcopter is flying at 1 to 1.5 meter altitude, but now it has the capacity to fly up to 500 meters altitude. You can see my students working on that, and you can see that the aerial presence of our quadcopter, where it is monitoring the atmosphere. And we have used it for monitoring the plant pathogens. This is the village next to Kanchipuram district in the state of Tamil Nadu in India. So our quadcopter is flying above the agriculture fields and different agriculture fields, and we want to study about the difference in fungal presence in different agriculture fields. Using this, we can easily forecast the disease and save the farmers from the loss. So as we are using UAV, we came across the problem related with the weight of the sample carried by the UAV. So we are supposed to reduce the weight. Then we thought, okay, why don't we design a sampler which has its own self-propelling system? Then we developed this particular air sampler, which weighs you around 35 grams. Again, it is having a propeller unit on its own, operated by battery, and again, using RC, that is remote control from the ground station, you can operate when the quad flies to the atmosphere. Then as we have designed, then we want the sampler to be multi-utility one. Then we thought, why don't we design the sampler which has the potency to monitor physical, chemical, as well as biological stuff. If we are using it for biological stuff, we can use a single shot sampling for isolation of actinomycetes, bacteria, fungi, and algae at a single flight. So we have designed multi-utility air sampler right here, and you are seeing just a diagrammatic one, a patent date to be filed, and it is under designing now. Right, so you may be knowing what all my contribution to the field. Right now, what I'm working, how to combat the airborne fungi in indoors. This beautiful thing we have it in our own culture. So we made it into science, although the knowledge is available for more than 5,000 years. Defending scientifically is difficult. So in India, this is what the major problem when compared with other countries. Other countries easily recognize, whereas in India, we are struggling hard to prove that the reason how you can combat the airborne fungi, right? Havans. In India, we call it as havans. In other countries, Western countries, they generally call it as smudging. Chemically speaking, you can call it as fumigation. Whereas as a nanobiologist, if you want me to speak about this, then I can say the role of nanoparticles, right? How the smoke looks like, plenty of fullerenes, even the carbon dioxide, and carbon dioxide possessing different chemicals. How it is going to fight with the airborne fungi. This particular stuff is having the capacity to become airborne. When this is becoming airborne, it is going to combat with that of airborne fungi. So there is going to be some interaction happening between us. physically what's happening. They form coalescing molecule. When they are coalescing, the size grows. When it is exceeding 550 micrometer, automatically the airborne particle settles down. So this is how my study, that is havens, are going to remove the airborne fungi in the atmosphere. The organic compounds present along with that of the material what I have used in havens, they will attack a fungal cell wall and they may cause fatality. That is, the viability of the fungi may be lost. That is the second principle what I'm thinking about. The third one, the reactive oxygen species present in the same carbon dioxide, they will react with that of wall component, or otherwise they have the penetration ability of the fungal wall, that is chitin, and penetrating through it, and it denatures the DNA of the fungal spore. Thus again the viability of the fungi can be lost. So this is what currently we are working about, and we are expecting the results to be on the expected line. So after getting the result, we may be good in publishing that. I'm dedicating this particular lecture to late Professor Yasti Tilak, who is rightly termed as father of Indian aerobiology, hailing from Pune, and he has produced more than 100 PhD scholars in the field of aerobiology. And next to him, late Professor B.P.R. Vittal, my mentor. Without him, I'm nowhere in the field of aerobiology. And the entire training of mine belongs to Vittal, and I'm dedicating this particular lecture to these two people. Before I finish, my thanks are to Dr. K.P. Kannan, the convener of this particular international conference. It is very difficult for this to organize this particular conference, that too by COVID time. My appreciation goes to you, Dr. Kannan. And my chairperson, Dr. Kumaresan, and co-chair Kriba for hosting me, the organizing secretaries, and then the participant, Dr. Sashireka is also here. My salutation to you, senior, and many members, and thanks everyone for listening, and your queries are welcome.
Thank you, sir. The participants can post their queries in the chat box.
In the meantime, I want to thank Dr. Uday, and it is an inspirational lecture. And I am very much thankful to you, and I am very much thankful to Dr. Kannan for organizing such a wonderful conference in this pandemic situation. So I want to actually summarize all those things. Senior, please mute your mic.
Sure.
Mic. Yes, thank you, Senior. So actually, nowadays the people not doing any taxonomical study, fungal taxonomical study. There is no people working in the field of aerobiology or mycetes or airborne fungi. So in those days, the great Professor B.B.R. Vittal is a great man, and I was also there in that time with my senior Dr. Uday Prakash. He's a pioneer in research in the field of aeromycology. So actually, the people come forward to work in the fungi and fungal taxonomy. Actually, in that time also, around 2003 and 2004, so many my seniors, they went for postdoctoral fellow for fungal taxonomy, especially in US. So there are a lot of opportunities are there to work in taxonomy, not only fungal taxonomy and also plant taxonomy, because people are working in, always they are working in molecular biology, cloning and other work. So it is also important, but then before going for molecular and other studies and technological studies, it is very much important to do taxonomical and fundamental survey study in the field of fungi and also in the field of airborne mycology. especially my senior Dr. Uday, fantastic lecture after long time, almost 15 to 16 years. Now I am hearing you, my senior, all the wonderful work you have done in the field of aromycetes. You are also a pillar of support for the aromycetes fungi group. They are working in so many people in the all over India. So, and also you proud that you are a student of Dr. B.B.R. Vittal. He's a great person. And also you have covered all the aspects in your lecture. That is the wonderful thing. Participant has to think about it. Actually, from the beginning, everyone, you thought about the fungal taxonomical work. He is expert. He is doing all those things. So from the fundamental to taxonomy to technology, how he is evolved, all kind of studies. So it is fantastic work. Most of the engineering college people not doing this kind of work, right? So he's basically he's a biologist, and he's done excellent work in the field of aerobiology, and also he developed some of the technology to monitor the air system or air pollutant, and also he summarized all the impacts, how it's affected agriculture, health, and environmental and other aspects, and also what is the medical importance. Even we are thinking about the COVID-19 virus, but maybe it is it will be there another 2 months, 3 months after the people, they will identify the vaccine. Then everything goes to the biopharmaceutical industry people. They'll earn money, and so many things are there in the background, but I don't want to disclose all those things. But then you think about the fungi, and slowly it will slowly do all kind of things to the environment, and also plants, animals, flora and fauna. Commonly we can say that flora and fauna. So and also the final thing what he said is carbon dot. So that is fantastic, right? So actually he is correlating with the ancient technology, decoding ancient technology with scientific proof. That is more important. So actually I appreciate my senior Dr. Uday Prakash. He's done excellent work in the field of aromycetes, and also contributed so many to the field of aromycetes. I thank once again Dr. Kannan and the department head and other colleagues in the Banari Institute of Technology, and all the participants. See, actually these kind of lecture you never seen in any of the forum. So because this is a rare field of fungi aromycology, so I thank once again all the participants have listen a good lecture, and also you have new idea to initiate to work in the field of aromycetes. I thank Dr. Kannan and Dr. Uday, and also co-chairperson. Thank you very much, my senior, Dr. Uday Prakash.
Yeah, thank you, Kumarazan. There are some questions in the chat box. There are a few questions are available in the chat box.
Yeah, Dr. Kannan, I have answered everyone.
In the chat level.
Yeah.
Senior, I'll read the question.
I already defended through chat box itself, Dr. Kumarazan.
Okay. So, okay.
Thank you, sir. I thank everyone who has attended the session, and the session will resume by 3:30. Thank you.